Hello, hi students. My name is Ushasri. I am working as JSO Biochemistry in Sports Authority of India, Bangalore. Today I am going to explain you about Nutrigenetics and Nutrigenomics. After watching this video, you would be able to understand what is Nutrigenetics and what is Nutrigenomics. Could nutritional genomics be applied to personalized sports nutrition? How we can use this in the field of sport? And what are the benefits of nutrigenetics and nutrigenomics. First, let us talk about nutritional genomics. It is a new branch of nutritional medicine based on the concepts of functional genomics and personalized medicine. In a simple way, we can say it is the study of how different foods can interact with a particular gene to increase the risk of diseases such as type 2 diabetes, obesity, heart diseases, and some cancers, etc. The main goal of nutritional genomics is to use personalized diets to prevent or to delay the onset of diseases and to optimize human health or to maintain human health. So, nutritional genomics is the study. It is about how food components or different nutrients that are present in the food will interact with different genes and how they alter the synthesis of proteins and how they alter the metabolites and how that is going to affect the health of the individual. At the genetic level, two terms, nutrigenetics and nutrigenomics, have been developed to indicate subfields focused on the intricate relationships between nutrients, genes, and biological systems. Nutrigenetics aim to analyze the impact of genetic differences between the individuals on the response to dietary intake and ultimate influence on health status and disease risk. So, it aims to analyze and understand how the genetic background of an individual or genetic polymorphism can modulate nutrients absorption, distribution, metabolism, and elimination. On the other hand, nutrigenomics integrates genomics and nutrition. It mainly focuses on the interactions between dietary components and the genome and the changes which takes place in protein synthesis and metabolite production and how dietary interventions can affect the gene expression and what are the best strategies of dietary intervention to prevent diet-related diseases, it explains. So, nutrigenetics and nutrigenomics are like two sides of the coin. Nutrigenetics explains how the individual genotypes will affect the utilization of nutrients. On the other hand, nutrigenomics study about how nutrients that are present in the food will regulate the gene expression. You must have heard about the popular proverb, one man's food is another man's poison. Every individual will have specific genetic content. Genetic variation is very common among the individuals. Genetic variation is the difference in DNA among the individuals. There are multiple sources of genetic variation, including mutation and genetic recombination, etc. Genetic variation is important in evolution. So the genetic variation is what makes us all unique. Single nucleotide polymorphism, also called as SNPs or SNPs, are the most common type of genetic variation among the people. Single nucleotide polymorphism is a DNA sequence variation occurring when a single nucleotide, adenine, thymine, or guanine or cytosine in the genome differs between members of a species or paired chromosomes in an individual. For example, if you see, these three individuals are having almost similar type of genetic sequence, but this first man is having adenine, and the second person is having guanine, and the third person is having thymine in the same place. So they are having different genetic content based upon one nucleotide. This is the best example of single nucleotide polymorphism. Let's recall central dogma before getting into nutrigenomics in detail. Central dogma is the process in which the genetic information flows from DNA to RNA to make a functional protein. As you all know, new DNA will be formed from the existing DNA by a process called replication. And synthesis of RNA from DNA template is called as transcription. And formation of protein or synthesis of protein from RNA is called as translation. So every individual will have unique genetic content. Based on this information which is present on DNA, specific RNA will be synthesized, and this information which is present in RNA will decide the protein that is going to be produced. Most of the nutrients regulate transcription and translation and results in altered protein synthesis. This protein could be structural protein, or it can act as enzyme, it may be an hormone. So finally it will elicit the metabolic response. So food is more than calories, and food is equal to information. Nutrients that are present in your food will alter the genetic expression either directly or indirectly. Let's see how nutrients acts as regulators of gene expression. Nutrients that are present in food will alter the genetic expression, means production of mRNA from DNA and formation or synthesis of protein from mRNA. Due to this, the concentration of protein or production of protein will be fluctuated, and this protein will elicit the final biological response. That could be metabolism, that can be cell growth or differentiation. So here nutrient is regulating the gene expression, it is regulating the protein synthesis, and it is regulating the metabolism, cell growth and differentiation of the organism. Let me explain how cholesterol regulates its own metabolism through gene expression. As you all know, cholesterol is very important for growth because it is a component of cellular membranes, and it is also precursor for steroid. Cholesterol uptake by the cells is mediated by a receptor called LDL receptor, low density lipoprotein receptor. So when the cholesterol concentration is low in cells, that increases the release of sterol regulatory element binding protein from the endoplasmic reticulum and Golgi. This will go and bind with the sterol regulatory element of DNA. This increases the genetic expression. This increases the formation of mRNA, transcription of mRNA and translation. So protein production increases, protein of transport receptor. So this increased protein production increases the receptor numbers on the cells. So more amount of cholesterol can enter into the cells. Along with that, decreased cholesterol levels also increases the enzymes that are involved in the synthesis of cholesterol. So when the cholesterol levels are low, cells will produce more amount of receptor proteins in order to increase the uptake of cholesterol into the cells, and cells also increases the enzymes that are involved in the synthesis of cholesterol so that more cholesterol can be produced inside the cells. On the other hand, when the cholesterol levels are high, less amount of receptor will be produced, receptor production decreases, and enzymes that are involved in synthesis of cholesterol also decreases. This is how cholesterol levels will bring back to the normal level. Here is a list of bioactive food components that can alter the gene expression. That includes micronutrients like vitamins and minerals, macronutrients like fats, carbohydrates and proteins, and other foods like flavonoids, polyphenols, xenobiotics, etc. Why is the interest? Why nutrigenetics and nutrigenomics are gaining more attention? Because this can be used to improve the health of the populations. This can help us to improve the athletes' performance, and it can be used to increase the weight loss in obese people, and there is a potential economic impact of. Could nutritional genomics be applied to personalized sports nutrition? Yes, One of the most effective applications of nutritional genomics can be in the athletic performance. Nowadays, athletes who are playing different games must conjugate physical activity with the personalized nutritional regime in order to maximize muscle growth and athletic performance. The genetic variability between the individuals can affect the muscle strength, skeletal structure, heart and lung size, tendon elasticity, etc., leading to different human phenotypes, ultimately influencing the sports performance. So, nutrigenomics testing includes several micronutrients that have modified requirements due to genetic variation, such as vitamin A, B12, C, D, E, calcium, iron, choline, and other bioactive substances such as caffeine. So, nutrigenomics works by identifying genetic variations that can help to determine the individual nutritional requirements. And it is also helpful to detect the presence of food intolerances, sensitivities, and optimal dietary patterns that will help to improve the health, body composition, and sports performance of athletes. Personalized nutrition based on an individual's genotype is not a new concept. There are several examples of genetic disorders like phenylketonuria, lactose intolerance, celiac disease, etc., where specific dietary recommendations are implemented to manage metabolic deficiencies. If you see the lactose metabolism, lactose is a sugar present in dairy products. It's a disaccharide. It contains glucose and galactose molecules. When lactose is injected through the diet, it will be digested by an enzyme called lactase. Lactase digests the lactose into glucose and galactose. But in few people, with the age, lactase levels decreases. Because of this reason, lactose cannot be digested. Due to bacterial fermentation, lactose produces gas and acids, and this results in flatulence, abdominal pain, etc. Phenylketonuria is another rare genetic disorder where the person cannot produce an enzyme called phenylalanine hydroxylase. This is the enzyme that converts phenylalanine into tyrosine. Due to the lack of phenylalanine hydroxylase, phenylalanine levels build up in blood and it results in many health problems. Let us see how vitamin A regulates gene expression. As you all know, vitamin A is very important for its antioxidant roles. It is also important to maintain eye health, which is critical asset in most of the sports where hand and eye coordination is required. Vitamin A is present in three forms, that is retinol, that is aldehyde form, retinoic acid that contains COOH group, and retinol that contains OH group. Vitamin A that is obtained from plant sources, mostly present in beta carotene form. Beta carotene is the precursor of vitamin A, or we can also say provitamin A. When we consume beta carotene from plant foods, this will be converted to two molecules of retinol with the help of an enzyme called beta carotene monooxygenase 1. Beta carotinase monooxygenase 1 is coded by a gene BCMO1. This conversion of beta carotene to retinol mainly occurs in intestinal mucosa and also in liver and some other organs. Every person will have 2 copies of BCMO1 gene, but about 45% of the population carries at least one gene variation that reduces beta carotene monooxygenase 1 enzyme activity. This significantly impairs the conversion of beta carotene to retinol. So, testing of BCMO1 gene will give information about the ability of the person to convert beta carotene to retinol. On the other hand, vitamin A in the form of retinoic acid also regulates gene expression. All trans-retinol and 9-cis-retinol are transported to the nucleus of the cell with the help of retinoic acid binding proteins. Within the nucleus, trans-retinoic acid binds with retinoic acid receptor, and 9-cis-retinoic acid binds with RXR receptor. After binding with RA ligand, RAR and RXR forms heterodimers which bind to the regulatory regions of chromosome called retinoic acid responsive element and regulates the rate of gene transcription. Vitamin A affects cell differentiation, maturity, and immunological function. Another important vitamin that is well studied under nutrigenomics is vitamin D. Vitamin D is present in food in the form of cholecalciferol, and it is available in plant foods in the form of ergocalciferol. We also produce vitamin D when we are exposed to sunlight due to the conversion of cholesterol into vitamin D in skin. Cholecalciferol is inactive form of vitamin D. It has to undergo 2 hydroxylation reactions to make calcitriol or active form of vitamin D. First hydroxylation reaction occurs in liver. Second hydroxylation reaction occurs in kidneys. And transportation of calcitriol in blood will happen with the help of a protein called vitamin D binding protein. It is also named as GC, group specific component. In cells, vitamin D binds with a nuclear receptor called VDR receptor, vitamin D receptor. It modulates over 200 genes that transcribe proteins that are involved in maintaining bone health, calcium homeostasis, muscle function, mitochondrial antioxidant systems, autoimmune regulation, synthesis of steroid hormones, etc. About 200 polymorphisms in vitamin D receptor gene are known, and others have been identified to be involved in vitamin D metabolism. Therefore, it is very easy to go into vitamin D deficiency for genetic reasons. The expression of VDR decreases with age, and VDR genotype is associated with lean mass, strength in older men and women. There is a strong correlation between vitamin D status and muscle function. So, hypovitaminosis D is associated with decline in muscle function. So, in nutrigenetics, 2 genes, namely CYP2R1 and GC, are analyzed to determine the risk of vitamin D deficiency. Variant of CYP2R1 can decreases the production of active form of vitamin D, and variant of GC protein or GC gene results in decreased transportation of vitamin D in body. In both the cases, deficiency symptoms can be seen. This diagram shows the role of vitamin D in gene expression. Vitamin D binds with the nuclear receptor called vitamin D receptor and alters the expression of many genes. As I told, around 200 genes are regulated through VDR receptor. Another compound that is well studied in the field of nutrigenomics is caffeine. Caffeine is one of the most widely used performance-enhancing drugs among athletes. The performance-enhancing effects of caffeine have been known for over 100 years and are well replicated in both endurance-based activities and in repeated high-intensity efforts. The ergogenic effect of caffeine is related to several different mechanisms. One of the known mechanisms is by inhibiting adenosine receptors. Caffeine competes with adenosine to bind with the adenosine receptors. This inhibition results in increased neurotransmitter release and muscle firing rates, an increase in adrenaline secretion and cellular ion release. It also decreases pain perception. If you see the caffeine metabolism, about 95% of the caffeine that is ingested is metabolized with the help of CYP1A2 enzyme encoded by a gene called CYP1A2. A single nucleotide polymorphism within CYP1A2 gene affects the speed of caffeine metabolism. Individuals with AA homozygotes are considered as fast caffeine metabolizers, and individuals of AC and CC genotype are considered as slow caffeine metabolizers. So, recent research shows that only people who are having AA genotype will be benefited by the ingestion of caffeine, but not AC and CC genotypes. Recently, Gerst et al. Demonstrated that caffeine consumption will improve the athletic performance only in AA genotype individuals, but not in AC and CC type individuals. In fact, athletes who are having CC genotype have shown diminished performance after the consumption of 4 mg per kg weight of caffeine. So, testing of CYP1A2 gene as a part of nutrigenetics will be helpful to know the genotype of the individual or an athlete and to see whether really caffeine consumption will be beneficial for them or not. Nutrigenomics testing addresses ideal macronutrient ratios, which can also tell you if your ideal body composition would be more easily achieved and maintained with a lower fat diet or a higher protein diet depending upon your genotype. For example, if you see FTO gene is also called as fat mass and obesity associated gene. It plays a role in the regulation of lipolysis and has been widely reported to be associated with fat mass and obesity in both adults and children. Gio et al. In 2015 has investigated the association of FTO polymorphism on the effects of diet and exercise intervention. At the pre-intervention time, individuals with A risk allele, that is AA and ET genotype, had higher BMI compared to those with TT genotype. But interestingly, biochemical and BMI parameters were significantly reduced after the intervention only in AA and AT genotypes, showing that the effect of exercise combined with diet intervention in Chinese children and adolescents is more effective for certain individuals depending on the genotype. Nutrigenomics testing help us to use genetic information to personalize a nutritional regime and to maximize the performance of the athletes. Along with that, nutrigenomics testing has also been shown to enhance motivation and behavioral changes. Genetically customized diet therapy is provided in India also. Herewith, I am giving the details of few of the consultancies. First one is Gene Support, that is located in Pune. Gene Support Wellness Private Limited is a private company, provides a wide-ranging test panel to health-conscious individuals. Chief among them is the DNA diet plan for weight management. The second one is Swagene. It is located in Chennai. Works at identifying from genes exactly which medication, surgeries, procedures will help to regain the health. They test genes and give personalized suggestions for diet and lifestyle, beauty, nutrition, and for fitness and excellence. Map My Genome is a molecular diagnostic company located in Hyderabad and Delhi. It offers personalized health solutions based on genetic tests. By combining genetic health profile and health history with genetic counseling, Map My Genome provides actionable steps for individuals and their physicians towards a healthier life. Benefits of nutrigenomics include increased focus on healthy diet and lifestyle, motivate positive behavioral change, increased awareness of risk of certain conditions, improved health and quality of life. It focuses on prevention of many diseases that decreases morbidity and premature mortality and reduces healthcare costs. Nutrigenomics helps to identify subgroups who might be particularly responsive or resistant to environmental or dietary intervention, and it is also helpful for better understanding of mechanisms involved in disease susceptibility. In summary, nutritional genomics technologies can be integrated with databases of genetic sequences, inter-individual genetic variability and disease susceptibility, etc. By this knowledge, we can elucidate the role of nutrients on various diseases such as hypertension, cancer, cardiovascular, and other life-threatening disorders. Thank you.